I'm Paola Vergani. There's my email there. If there's anything in this lecture that is unclear, yeah, we should start. So I'll be talking about drug discovery in general. We'll start from how this was done in the past. We'll introduce some aspects of modern drug discovery, and we'll talk briefly about an aspect of drug discovery that is likely to become more and more important in the future. You'll have more on modern drug discovery by Professor Whiting in the next lecture. So this is sort of an introduction. So animals, fungi, living organisms in general produce millions of different natural compounds. And a small number of these are actually beneficial for human disease. Use of natural compounds to treat humans is a very ancient practice. It has been done for thousands of years. Examples of natural compounds that have effects on humans are given there. So morphine is extracted from the opium poppy, and use of the opium poppy is mentioned in a papyrus dating back to 1,500 years before Christ. Another compound that is useful for the treatment of disease is digoxin. It is extracted from the plant, the foxglove, and it's used to treat heart conditions. Morphine is used as a painkiller, as well as for recreation. One big problem with the use of natural products is that very often they were used as extracts. And these extracts contain mixtures of many different compounds in variable concentrations, uncontrolled concentrations. So in recent years, we still use natural remedies, but the active component has been identified, purified, and is used in controlled concentrations. In the past, drugs were discovered mainly by chance. And the most famous drug discovery by chance is the discovery of the antibacterial properties of penicillin, made by fungi of the genus Penicillium. This discovery was made in the 30s by Alexander Fleming, and it's simply a chance discovery. So he had some Staphylococcus cultures, and he just had one culture in which he observed something similar to that, what is shown there. So actually, maybe I should reduce the lighting. Okay, so you can see a bit better. The colonies of Staphylococcus growing far away from mold contaminants are still normal. They grow normally, but growth close to the mold is inhibited. And this identified, so told Fleming that there was something in the secretions of that mold that was inhibiting staphylococcal growth. Careful analysis of the mold and the compounds led to the development of penicillin, which has obviously been of great importance in medicine. However, modern drug discovery drug that can be thought of as modern drug discovery is the effort led by Paul Ehrlich at the beginning of the 1900s to discover a drug that could treat syphilis. What they knew at that time was that arsenilic acid could kill the trypanosomes that caused syphilis. However, this compound was extremely toxic to humans. So what Paul Ehrlich and his colleagues did, in particular organic chemist Alfred Wertheim, they synthesized a very large number of compounds similar to arsenilic acid and tested each one for biological activity. So when they got to compound number 606, they finally found what they were looking for. They found salvarsan, this compound shown here, and this had the properties that they were looking for. It was still able to kill the trypanosomes, so to treat the syphilis, but the concentrations that were toxic to humans were much lower. So it had a much better therapeutic window. At concentrations at which it killed the trypanosomes, it was not terribly toxic for humans. So this is the first time in which there was a team effort to, starting from a lead compound, to systematically produce different structures related to this lead compound and test each one until the desired biological activity was found. Yeah?
What date was Paul Ehrlich?
This was at the beginning, I think salvarsan was discovered in 1910.
And the 605 chemicals that didn't work, who were they testing that on?
They were testing them on, well, they had some assay for humans. They were not testing them on humans. I don't know exactly what the arsenilic acid was toxic for, but they had some assay. Probably at that point they might have been testing it on mice, some animal model, but I'm not sure actually. Good question. So obviously the bacterial action on the syphilis-causing bacteria could be tested in vitro, but you're right, yeah. So this is the second example of structure-activity relationship study. So salvarsan was also obtained through a SAR, structure-activity relationship. Again, we start from a lead compound, modify it chemically, and test until we find what we want. In this case, Sir James Black, he was working on angina, a condition in which the heart has insufficient oxygen for its needs. And Sir James knew that activation with adrenaline and noradrenaline of adrenal receptors increased the heart rate and the strength of contraction of the heart. So he thought if he could get something to block the action of adrenaline, this would reduce the oxygen requirements of the heart and would be beneficial in angina. So he started his lead compound, in this case, was an agonist for the adrenal receptor. Adrenal receptors have roughly two classes, alpha and beta, and in the heart you have beta-1 receptors. Okay? So he wanted something to block the beta-1 receptor, but what he started with was a beta agonist, isoprenaline. It was also known that dichloroisoprenaline was a partial agonist. So it was also known that just this change of the OH groups for chloride made the agonist into a partial agonist. Again, Sir James changed the structure out and tested the resulting chemicals, and this led to the discovery of propranolol, which was the first beta blocker that was used in humans. So again, structure-activity relationships, SAR. Now we can just move to classifying how modern drug discovery is carried out in pharmaceutical industries today. Broadly speaking, we have two steps. We have a first step, that is the identification of a target and a lead compound, and then a second step in which this lead compound is optimized. So let's start with the first step, identification of a target and of a lead compound. So what are targets for drugs? Receptors, right? But what are receptors? Proteins. In general, they're proteins. So you can have receptors that are channels or integral membrane proteins. They are also various kinds of enzymes or transporters or structural proteins. Any kind of protein can be a target for a drug to cure some disease. And mainly, target identification is based on biological intelligence, what we know about that specific disease and the molecular mechanisms of that specific disease. And very often, lead identification is also obtained through biological intelligence. So, the second step is involved in optimizing this lead compound. You need to minimize the negative effects and maximize the wanted effects. So you want to increase the potency on the target. You want to have it as low a concentration effective as possible. You want to improve its pharmacokinetics. So you don't want something that builds up in the body or that is not metabolized at all, or that is metabolized too quickly, for example. So you have to optimize the compound in order for it to be reaching the target and diffusing through the body and reaching the target and not building up to toxic concentrations. And minimize these unwanted toxic effects that occur usually when the drug binds to proteins that are not the target. So let's look at examples. The first is an example of target and lead identification through biological intelligence. So we are in the 70s, and at that time it was known that cholesterol synthesis occurred mainly in the liver in humans, and one step of cholesterol synthesis was catalyzed by this enzyme, HMG-CoA reductase. And it was also known that a high level of cholesterol was associated with an increased risk of cardiovascular disease. So Akira Endo in Japan and his co-workers reasoned that since mevalonic acid, the product of this reaction, is also an important precursor in the biosynthesis of the cell wall and the membrane components of microorganisms, they thought that probably there was some microorganism that had evolved an inhibitor of this enzyme to fight other microorganisms in the fight for survival. So what they did was they had an in vitro assay in which they could test the effect of extracts from various cultures of microorganisms on this enzyme, on the activity of this enzyme, and they identified finally a drug called nevastatin, who was able to inhibit HMG-CoA reductase. It was synthesized by this fungus, Penicillium supremum. So here we have identification of the target based on the knowledge of synthesis of cholesterol in the human, and identification of a lead based on microorganism ecology, both biological intelligence. Now, they identified the lead starting from extracts from microorganisms. Very often, drug companies don't have a way of selecting compounds that are more likely to be active on the market. So what they do is to test a very, very large collection of random compounds that have characteristics that could be made into drugs. So these are compound libraries of millions of compounds. But in order to be able to test these, the drug companies have to develop assays that can be automated, that can be run quickly, and so that you can test thousands of compounds a day, millions of compounds over years of research. This screening is called high throughput screening, HTS, automated screening. So an example of this is an assay that was developed to identify a drug to treat thrombocytopenia. This is a condition in which there is a low platelet count in the blood. And it's often found in patients that are undergoing chemotherapy, serious chemotherapy for cancer. So the drug target in this case was the thrombopoietin receptor. This receptor, physiologically, is present in the stem cells in the bone marrow, and activation of this receptor triggers a signal transduction cascade that results in the activation of specific genes and therefore the differentiation of platelets. So these genes that are activated or inactivated actually contain a promoter that has an element that is responsive to activation by the thrombopoietin receptor. Okay? And what the scientists in GSK did was to express the thrombopoietin receptor in a cell line, and in this cell line they also put a synthetic gene in which they put this thrombopoietin receptor activation responsive promoter upstream of a gene encoding a luciferase. Luciferase is an enzyme that catalyzes a reaction that emits light. Okay? So if you can imagine that you can seed these cells expressing both the receptor and the synthetic gene in these miniaturized wells, like 96 well plates or 384 well plates. And then you can automatically read the light that is emitted in each well. You can identify compounds that are able to activate the TPO receptor, and this will result in transcription of the luciferase and therefore emission of light. So only if the well receives a compound that is an agonist for the TPO receptor, will the luciferase be transcribed and therefore will light be emitted. So assays like this, so here the readout is the emission of light, and you can have robotics that add the various cells. The assay can be automated so that tens of thousands, hundreds of thousands of compounds can be tested every day. So in this case, 260,000 compounds were tested, and a lead was identified that was then optimized into a drug, L-Propag, that is now still used by patients to treat patients. So high throughput screening is a way in which most lead compounds are identified nowadays. However, our biological intelligence has increased a lot. And because of a lot of effort in research labs and in academia and in industry, we have the atomic structure, crystal structures, of many proteins that are medically relevant. So if your target, if the structure of the target that you have chosen is known, you can select the compounds to test based on this biological intelligence of knowledge of the structure. So, in this case, the scientists were looking for a drug to treat leukemias, and the structure of tyrosine receptor kinase, a receptor tyrosine kinase, involved in the development of a large number of this kind of leukemias was known. The drug was designed to fit the active site of the tyrosine kinase. In other cases, you have other functional cavities in the structure that are functionally important. But so, with our knowledge having advanced so much, it becomes evident that if we use the shape of the protein in order to design the drugs, individual variations in the shape of this protein might result in different responses of individual patients to the same drug. So genetic differences that cause target proteins to have a slightly different shape might result in a difference in response to drugs by different individuals. And that leads us to the last part of this lecture, personalized medicine. So we can start off from the observation that if a group of people, of patients, all have the same diagnosis and are given the same treatment, not all of them will respond in the same way. So you will have some of them that will benefit from the treatment and others that will not benefit from the treatment. Some of them will experience adverse drug reactions, and some of them will experience no toxicity. So different people, individual people, will have different responses to the same treatment. Studies have shown that the variation in drug response is largely determined by genetic differences in the individuals. So the color in this diagram is mainly determined by the genotype of the patient. And pharmacogenetics is, in fact, the study of how these genetic differences result in different responses to drugs. Now, studies have shown, so we have a few examples in which we understand how the genetic difference is linked to the different response. And these fall in two main classes. So one class is genetic differences in genes that encode proteins that are involved in the pharmacokinetics of the drugs: absorption, distribution, metabolism, and excretion. So if you have individuals that have differences in genes that are involved in how the body handles the drugs, then knowing the genotype of the individual patients will allow the doctors to select the right drug concentration, the right dose to give to each individual. The second class of genetic differences resulting in differences in drug response are differences in the gene encoding the target of the drug, or proteins directly linked in the signal technology chain. So here, the genetic differences actually cause differences in shape of the targets. So in this case, genotyping patients would allow doctors to select the best drugs, the drug that is most effective for that patient, rather than the best dose, select the actual best drug, most effective drug. So here we start with the first example. This is an example of a genetic difference in a pharmacokinetic enzyme. So succinylcholine is a skeletal muscle relaxant. It's an agonist at the acetylcholine receptor, postsynaptic. And usually when patients are treated with succinylcholine, they can recover activity of their muscles within 10 minutes. And this is because the succinylcholine is metabolized by an enzyme called butyrylcholinesterase that is present throughout the body in the plasma. So this is what happens in the vast majority of patients. In 10 minutes, the succinylcholine is metabolized. But in a small proportion of patients, these have an allele that encodes different polymorphisms. So instead of having an aspartic acid at position 70, they have a glycine. And if this particular allele is present in the homozygous form, patients take up to 10 hours to recover. So this is obviously very, very damaging, and we don't want this to happen. So patients are screened before they're given succinylcholine for this particular allele in the butyrylcholinesterase. A second example of genetic variation leading to differences in drug response. Here we start off from a large clinical trial in which statins were being tested. And then you have 6,000 patients that are given statins, and 6,000 patients are given placebo, and the aim there was to see whether statins truly had an effect on the risk of cardiovascular disease. among the 6,000 patients that were given statins, 85 of them developed adverse drug reaction. They developed some muscle soreness and myopathy that is related to the statins. 90 controls were chosen to match these 85 patients. These 90 controls belonged to the 6,000 that were given the statins in the first place. And for each of these 85 case and the 95 control individuals, scientists genotyped them so that they could measure, they could determine the alleles present in more than 300,000 SNPs. SNPs, single nucleotide polymorphism, distributed throughout all the chromosomes, the 23 chromosomes. So SNPs are the smallest element of genetic variation, single nucleotide polymorphisms. Okay? So each one of those dots is an SNP, a different SNP. And on the y-axis, you have association with myopathy. The higher the dot is, the less likely this association is to occur by chance. Okay? So for most SNPs, you have a very low association. But one dot, that black dot up there, has a very strong association with myopathy. So the probability of having this association by chance is roughly 10 to the minus 6. Okay? So this was unprecedented. There was one SNP, one particular genetic variation that was strongly associated with the risk of having this myopathy. This gene was in, this SNP was in a gene encoding a transporter that is expressed in the liver and that is involved in the uptake of statins in order for them to be metabolized. So it's again an example of a protein that is involved in the pharmacokinetics of the drug. So having this particular genetic variant greatly increased the risk of myopathy for the patients. Yeah?
Does epigenetics influence also genetics?
Epigenetics, what do you mean by that? These are differences that are not encoded in the genome, or?
Yeah, and the expression of genes.
Probably there will be some genetic variation that is determined by epigenetic individual characteristics. But in most cases, some genetic underlying cause is found, and that seems to be dominant. It's probably true. We'll probably in the future find out that there are actually also epigenetic differences that cause differences in drug response. Yeah.
The reason why they
I'm not sure. I think actually X and Y. I think that means X and Y. They just mentioned X because it's the more complete. So I'm not sure. I can look it up for you. So the next examples are in proteins. So the target protein is the one that has the genetic differences in this case. So HER2 is a receptor tyrosine kinase that is involved in the cell cycle, in regulation of the cell cycle. And it's overexpressed in 20% of breast cancers. So activation of the receptor results in cell proliferation. A new drug was designed. This is a monoclonal antibody against the ligand binding domain of HER2, binding of this drug to HER2 prevents dimerization and therefore prevents activation of the tyrosine kinase. And therefore it results in decreased proliferation and actually also apoptosis. Now, how can we determine whether a woman with breast cancer would benefit from treatment with this monoclonal antibody? This is done by testing of the cancer. A small biopsy is removed from the cancer and is tested. In this case, you have immunocytochemistry. You have the nuclei that are stained blue, and the HER2 is stained brown. And you can see that this first patient here does not have overexpression of HER2. So they are not likely to benefit from treatment with Herceptin. This patient here has a very strong expression of HER2. And therefore, they are the ones that are chosen for treatment with Herceptin. So this is a first example of genetic difference in the target protein that determines response to a drug. In this case, it's overexpression of the target protein rather than actually the shape of the target protein. But the last example is actually regarding the shape of the target protein. So cystic fibrosis is a genetic disease, and it's one of the most common genetic diseases. And it's caused by a mutation in the gene that encodes for CFTR. CFTR is an anion channel that is expressed in various epithelia, including the airways and the guts. It's expressed at the apical membrane in these polarized cells. We have some idea of how the CFTR protein looks, the shape of the CFTR protein. So this one is actually a homology model. It's based on crystal structures of bacterial homologs. But actually just last week, a structure of a CFTR protein has been solved. So I should change this slide, but I didn't have time to do it so far. So it's not very, very different. We also know not only the shape of CFTR, but we also know the defect present, the mutation present in patients. The most common mutation is deletion of F508, that pink, shown in pink in space down up there, in the center of the protein. And so more than 90% of cystic fibrosis patients have at least one allele of delta F508. So it's really the most common mutation. The second most common mutation is G551D. So a glycine is substituted by an aspartate in a crucial point at the center of those nucleotide binding domains. This mutation is present only in about 4% of the patients. So it's the second most common, but it's much less common than delta F508. We also have an idea of the molecular defects caused by each of these mutations. So delta F508 has a strong effect on the folding of the protein. So the protein is trapped in the ER, it can't fold properly, and it's targeted for degradation. Most of it never leaves the ER. A little proportion does leave the ER and is trafficked to the plasma membrane, but there is a gating defect. The channel, instead of opening and closing, like the normal CFTR, it essentially stays closed all of the time. So the open probability is greatly reduced. It has defective folding and defective gating. The G551D mutation only has the defective gating. So it's folded, it's trafficked to the apical plasma membrane properly, but once it's at the plasma membrane, it stays closed all of the time. It never opens. It opens very, very briefly and rarely. Now, recently, after a high-throughput screening assay was developed, a new drug that could activate CFTR, that could increase the open probability, could make the channel stay open longer, was developed. This is called VX770 or ivacaftor or Calideco, many different names. And this has been life-changing. So treatment with this drug has been life-changing, but only for patients that carry the G551D mutation, at least on one allele. Other mutations, so there are like more than 1,000 mutations that cause cystic fibrosis, and recently another set of gating mutations have been shown to be that can be treated with ivacaftor. So it's not only G551D, but it's still about 5% of the CF population that can be treated with this new drug. However, treating clinical trials in which patients carrying delta F508 alleles were treated with ivacaftor didn't show any benefit for the patient. In fact, trials have been also carried out using other drugs that were supposed to affect the folding and affect the folding of delta F508 in vitro, but they didn't give any benefits in clinical trials. And also trials in which VX770 ivacaftor with these correctors, these folding correctors, were given together, they also gave extremely poor responses in CF patients with delta F508. So we are currently working on developing drugs for delta F508. But this is a specific example in which the target protein, you have genetic differences in the patients that determine the response to the drug. In this case, it's an extremely simple difference. So there is only one gene that causes the disease, and polymorphisms in that gene determine whether you respond or you don't respond to the drug. So it's a relatively simple case. But actually what has emerged is that in most cases of drugs and diseases, the response is not determined by polymorphisms in a single gene, but the situation is much more complex. So, to summarize, in the future, a lot of pharmacological research will be done to determine whether drugs have different effects in different people, differences that are both in the efficacy of the treatment, so some treatments might be efficacious for some genetic variants and not others, and in the toxicity of the treatment. These pharmacogenomic studies. Why have we now gone to pharmacogenomics rather than pharmacogenetics that we started off with? This is because, as I said, we are realizing that the situation is not simple. And we can't usually link individual genes to genetic variation, causing variation in response to drugs. But it's more sets of polymorphisms that determine the drug response. So it's not single gene polymorphisms, but sets of SNPs. So with the increase in knowledge that we are slowly building up, and with the increase in technology that makes genotyping, so determining sequences, cheaper and cheaper, it is likely that individualized or stratified medicine will become more and more important, rather than talking about having a drug, having patients each individually treated. We talk more about stratified medicine. Patients are divided, are classified in strata, having similar genetic characteristics. So in the future, this will probably become more and more important. At the moment, it's really only important in very few diseases: cancer, cystic fibrosis, leukemia. Okay, so does anybody have any questions? You'll get much more in-depth coverage about modern drug discovery in your next lecture. This was just sort of an introduction.